basically induced even more calcium release into the cytoplasm. And 90% of the calcium used in contractions are found in the sarcoplasmic reticulum, but you still need that extracellular calcium to induce the release of the SR calcium. And then the rest of it works pretty much like a skeletal contraction here. Calcium will bind onto troponin. Troponin then moves tropomyosin out of the way. Assuming there is ATP, you're going to get cross-bridge cycling here, and then you're going to get contraction here as you get a cross-bridge cycling event to occur. That part should be hopefully pretty obvious to you how it works. And then relaxation, so this right-hand side shows relaxation, works in a very similar way to skeletal muscles. Calcium gets transported back up through calcium ATPase, and calcium also gets transported out of the cell via the calcium sodium antiporter or exchanger. You could use those words interchangeably. So basically what it does, it sends calcium out, brings sodium in. That's what this guy does right here. So if you keep on doing that, you're going to get a whole bunch of intracellular sodium. Is that good or bad?
Not necessarily good.
I guess it depends on your view of good or bad. That's irregular. Generally you want to keep sodium outside the cell, right? We use sodium for a lot of things. We use sodium for action potentials. We use sodium for glucose transportation. We use it for a lot of things, so it's good to keep it outside. And so you need another exchanger. You need a potassium sodium antiporter or exchanger. You could write either word to then take sodium out, potassium in. That's a good thing because you want to hoard all the potassium inside the cell, and you want to keep sodium outside the cell. And so that's how a cardiac muscle cell will relax then. Bring calcium back into the SR, send calcium out of the cell, but in exchange you have to bring sodium in, but then throw sodium out by bringing potassium in. So I can ask a question like this. What if the sodium potassium ATPase malfunction? It doesn't work anymore. None of them work anymore. What would happen to the cardiac muscle cell contraction? It would stop. It would stop, right? You can still get calcium out, right? So that would relax the muscle cell, but you couldn't get another action potential to go because you have too much intracellular sodium that's going to depolarize your cell. So you're not, it hyperpolarizes your cell. So you're not going to have action potentials occur. It's not going to get beyond that threshold value, remember? Alright. So, like smooth muscle, cardiac muscles can have graded tension, meaning from every twitch, for every single twitch, it can produce varying amounts of tension. So this side is tension. And so just think about this. Now tension is directly proportional to cross-bridge cycling. Right? And then cross-bridge cycling is also proportional to how much calcium you have, how calcium is present. Right? And lastly, calcium presence is proportional in this example. Okay, is proportional to, you know, extracellular calcium intake. So what cardiac muscle cells can do is they can vary how much extracellular calcium they take in. They can take in a lot. If they take in a lot of extracellular calcium, that is going to release a lot of sarcoplasmic calcium, sarcoplasmic reticular SR calcium. And then a lot of that calcium can then lead to a lot of cross-bridge cycling. That's what this graph is here for. And that will lead more cross-bridge cycling, leads to more tension, right? And so we'll talk about it a little bit later, but there are ways to modify how much calcium can be let into these cardiac muscle cells. And that's how you can get greater tension in a cardiac muscle cell. All right, that's all I'm going to talk about cardiac muscle contractions. Any questions about that? No? All right.
Do these have the same chemicals in them as the ringostriated muscles of your skeletal
You mean,
muscles?
do they have like the sarcomeres and whatnot? Yeah, they do.
Tighten.
Yep, they have sarcomeres. They have all the good stuff that go along with sarcomeres.
All the same.
Yeah, so yeah, cardiac muscles are striated because they have all these sarcomeres. They have it in series. They have myofibrils. They have all that good stuff. Yep. Alright, so let's talk about the electrical conduction. So we know these cardiac muscles need to have an action potential to depolarize them. So let's talk about how depolarization travels across the whole heart. How do these action potentials get across the heart? And so first, let me switch to a brighter color. That's good. Maybe I can get something a little darker. Let's try black. I think black's better, yeah. Alright, good contrast. So, the depolarization of a heart, so basically the starting of all these action potentials start up here, kind of at this tip of the right atrium here. This part here is called the SA node. Or, the long form is the sinoatrial node. And so the SA node are full of these cells called pacemaker cells, or autorhythmic cells. And so what they do is they have a fluctuating cell membrane potential. So they basically are the cadence makers. They basically start action potentials on their own at a given rate. And the rate they create action potentials is going to be ultimately your heart rate. So SA node depolarization frequency is the same thing as heart rate. And that makes sense, right? If they're the ones that start the signal, how often they start the signal will determine how often your heart's going to contract, right? Because as the cardiac muscle cells get the action potential, get the depolarization, they're going to contract. And so every time they get that depolarization event, they're going to contract. So it makes sense that how often these SA nodes depolarize will determine your heart rate, how often your heart beats. And so from there, the signal, the depolarization spreads. So if this was the SA node, it moves on to another node called the AV node. An AV node is just the atrioventricular node. And it moves through these paths called internodal pathways. You don't need to know that. That's something from anatomy. But what you see here is, from step 1 to step 2, as they're taking these paths to the AV node, we start seeing part of the atria, this pink part right here, starting to depolarize. So that's what that pink shaded or the purple shaded part means. Those are the part of the myocardium that are starting to be depolarized. Keep in mind, that doesn't mean they're necessarily contracting just yet, they're just being depolarized. And so once the signal makes it to the AV node, it goes down this path right here, called the bundle of His. And then at this point, the atria have been fully depolarized and they have contracted. And you can kind of see that here. They're kind of misshapen compared to what they were before. They kind of have this kind of scrunched up shape to show that they're physically contracted. Mechanically, they contracted. From here, the signal will then branch out into the left and right bundles. And at this point, the signal will move into these branching fibers called Purkinje fibers. This helps spread the depolarization across a lot of the ventricles. And so you see that through all these Purkinje fibers that kind of come off not only the left and right bundle branches, but here in the septum, but also on the sides here, that we're depolarizing all of the ventricle. So at the end of the electrical conduction path, ventricles should all be depolarized should all be depolarized. So what happens if the AV node cannot depolarize? What will happen to the heart contractions as a whole? If you somehow knock out the AV node, saying the AV node can no longer conduct the depolarization, what will happen to the heart's contractions? Will everything stop? Just the ventricles. You will still have the